second part of the notes is really more about the function of the cell membrane and how does the cell membrane actually form some sort of barrier. So when we look at how it forms a barrier, we're really looking at how is the inside of the cell different from the outside of the cell. And the first thing we actually need to do is really just understand how molecules move at all. And this has to do with the second law of thermodynamics. The first law is that energy and matter are neither created nor destroyed. So that's the first law. You can't make it, you can't destroy it. The second law is really that molecules are in constant motion, actually towards what's known as entropy or disorder. That molecules do not usually stay in one place. Now, if you're a solid and you're connected to each other, that's different. But liquids and gases, the molecules have enough energy that they're actually moving around. So the first idea is really this idea of constant motion, and what we associate with is that molecules are actually going to spread out. As they have that energy, they're going to be bouncing off each other and other things until they're equally distributed or equally concentrated. So right now we would say that there's a high concentration. Remember, this is the abbreviation for concentration, those brackets. Right now there's a high concentration in this bottom right corner. And the molecules are going to tend to spread out. And the idea of spreading out, so spread out is the movement down a concentration gradient, which is from high concentration to low concentration. And so that is the idea of diffusion. That molecules are spreading out from a high concentration to a low concentration until equally distributed. So examples of this include smells. So when I spray a can of Lysol and I can smell it as it spreads out. Mixing cream into a coffee, diffusion. Anytime we see something spreading out, that's going to have to do with diffusion. One of the things in biology, though, when we're dealing with the cell, is that they're going to spread out until they reach a barrier. And so molecules are going to diffuse or spread out until they hit this barrier. Great picture here, because we see the kinks in the tails, we see the different phosphate heads. And so as the molecules are spreading out, once they reach a barrier, they can't spread out anymore. However, we do want to know this membrane is selectively permeable. So certain things are going to be able to move through. So we have some rules about being able to cross the membrane. These rules down here apply to molecules that don't naturally diffuse. Molecules that diffuse easily across the membrane, meaning no energy and no help, are going to be your small nonpolar or non-charged. And the ones we're really going to focus on are O2 and CO2. And so those are going to be able to squeeze through here. Some very small lipids, like especially lipid hormones, will be able to squeeze through. But the main things we focus on are these two gases that are able to diffuse across the membrane. Everything else is going to bounce into that barrier and not be able to pass through, except that the membrane is not just phospholipids. The membrane is actually composed of a mosaic of things, including proteins. And so what we have here to look at are some other examples of transport. The first is going to be a channel protein. And channel proteins are going to be transmembrane. They cross the membrane, so we know something about the polarity. And they're going to be shape-specific, shape and charge specific. And they're going to allow molecules to pass through. One of the most important chain of proteins is an aquaporin, and this allows water to move. Water can't fit through this nonpolar region, so there are actually proteins that are designed to allow water to move very quickly. And so water can move through what we call osmosis. So we'll focus on osmosis in just a few seconds. But right now we want to know that there are carrier proteins based on shape and charge that allow, channel proteins that allow molecules to move. No real help needed, just kind of like a trash chute. Like here you can fit this way. A carrier protein is almost a little bit more like an elevator in that it's going to fit in and the protein is actually going to change shape. So the big thing we want to know about a carrier protein is that we have a conformational or shape change. It doesn't always use energy. Sometimes it's kind of like the induced fit where it fits and then it pushes it through. So it's not necessarily that it needs energy as much as it's a little bit more complex. These are usually going to be very specific. Next we have the idea, this is actually a picture of exocytosis. And this is an example where we are allowing something to exit. The reverse could happen. Endocytosis could actually be coming in. Now these are being secreted, but endocytosis is where we're going to be bringing it in. What we notice there is that we become surrounded with the plasma membrane or that we merge with the plasma membrane. And we'll talk more about exocytosis a little later when we talk about these secretory vesicles. For now, we just need to know that sometimes it's the protein that's doing the movement, sometimes it's actually a membrane releasing or bringing in. And so all of these are going to be passive transport. So we've got examples where there is no energy, and we move from high to low concentrations. And so that is really what's happening here in this column. Over here, we've got some slightly different rules. These are examples if we want to collect something. So if we want to collect or move from low to high, that's actually going to require what's called active transport. And active transport, you can kind of see in this picture, is going to use ATP, which we'll talk a lot more about later in the year. It's the cell's energy currency, what we use for energy. And so the term active transport really does tell you we're using energy. It requires active force. So you'll usually see this idea of ATP or energy, or sometimes you'll see that we're going from low, like here with the potassium, to high. One of the important things about active transport is that sometimes it's coupled, meaning I've got two things moving at the same time. And that can be what's called a symport, where two things are moving in the same, sym meaning same direction, or antiport meaning opposite directions. So big idea here, we've got the cell membrane, it's a barrier. It's stopping things from diffusing or spreading out. Once they hit the barrier, they can either sneak through or they can move through a channel. They can have a specific carrier that changes shape. They can be excreted or brought in using the membrane. Then sometimes we go against the concentration gradient and we move from low to high. That is called against the concentration. Picture pushing a ball up against gravity, up a hill. And we're going to see this idea of pumping a lot. So anytime you see the word pump, that's going to refer to active transport. So basically we've got this idea that we can move things across the cell membrane using specific proteins and either using energy or going with the concentration gradient. We do actually want to spend some time focusing on the movement of water. And so what we're usually going to see is that water is one of the main things that's going to move by diffusion. The movement of water across a semi-permeable membrane is called osmosis. This word refers only to the movement of water, nothing else. And what we're going to see is that water, because of its property to be attracted to things, when there's a lot of solute, so these little dots here represent any given solute. A solute is something that is dissolved in water. Anytime that we have the solute, the water is usually going to be attracted to it. As the water is attracted to it, the water doesn't move around very much. And what this creates is a gradient where there's more solute and basically less free water molecules. So the more solute, a good rule of thumb is that the more solute, the less room for water. Well, we actually have a word for more solute, and that's hyper, like high, solute, or tonic. So a hypertonic solution has a high amount of solute. And what that means is less free water molecules because they're attracted. Remember that idea of all the water molecules attracted around like a sodium ion? So over here we have a lot more free water molecules. So those free water molecules are moving around, that thermodynamics, and so the net movement of water is going to be to this side. More solute, less free water molecules. Over here, hypo means less solute, so there are more free water molecules. So the water moves from high to low, and that's why the water concentration, or that's why the water number rises over here. Once we reach equilibrium, this is when the concentration is equal on both sides. So you'll notice the solute didn't move. The solute, maybe it was salt, maybe it was sugar. The solute was not able to pass this selectively permeable membrane. So because the solute couldn't spread out, the water did. And what we have is not the same amount of solute on each side. But remember, concentration is kind of like a ratio, meaning we have the same amount of water to solute on both sides. And we do have this idea of net. Water is actually still moving back and forth. These free water molecules are still moving around, but they're moving back and forth kind of equally. So one thing that we would need to know now is that they are what we call isotonic. Like an isosceles triangle, isotonic means we are equal on the sides. And so we do need to introduce these words to our vocabulary. Hypertonic, hypotonic, just think hyper, lots of energy, so high. Hyper, hypo is down, and iso is equal. And so what we need to be able to do is predict what a cell will do when put in a solution. So if I'm put in a hypotonic solution means that the solution has less solute, more water, and this is where we compare the solution to the cell. So if the cell were hypotonic, that would be different. Here they're saying the solution is hypotonic. So because there is more water in the solution, water will move in, the cell will swell, and will actually burst. Now in a plant cell, we'll talk that cell wall provides the support. So you should be able to predict what's going to happen in each solution, and then later we'll talk about using an equation to come up with quantitative amounts.